Good day. So, welcome back to studying microbiology chapter 1, scope and history. But today we are not going to finish the chapter. We are going to answer the questions that are asked in this chapter as to revise what we studied and not forget. So, going directly to that, I think we are going to look for the questions that are the start first. Wait, it's going way too down. Okay, why are we so bad at this? Well, give me a second. I need to go down. Something happened and it scrolled up. Wait. So I just want to see if this one's going to, if the voice and everything is fine, and then we are going to start in a second. Right. Where is the voice? We don't need. Alright, it's working. So let me. Alright, so we are here. Why are we so at the far? I should have searched it before, but I didn't want to do that. First, I need to know what the chapter page here is. Dang. This is making me angry. Where are we at chapter 4? Why are we at chapter 4? Right, why are we? Oh no. What is this? Here it is. Why am I getting angry? Let's do it casually. Let's go back page by page. Guys, if you are getting bored, just skip forward and see where the video is fine and stop there. Okay, we are here at the start. So in this page there is no question. Then in the next, here these are few questions. So I'm going to see if I remember things or not. So describe the field of microbiology in terms of size of its subject material, and the nature of its techniques. So they are asking me to describe the microbiology in terms of what? The size of its subject material. Right, like they are telling me the how much does the microbiology covers. So first of all, the microbiology covers all the microorganisms, and not just the microorganisms, everything that's related to the microorganisms. Microorganisms are bacteria, viruses, fungi, archaea, protozoa, and did I miss something? Algae, viruses, yeah, these are all, and everything in the surrounding that is related to it, and any field that is related to it, like food microbiology, industrial microbiology, ecology, microbial ecology, like these kind of things which we already studied in the movement further, and yeah, genetics and these kind of things. So, and the nature of its techniques. So the basic techniques is the culturing of microorganisms and the gram staining. here we studied somewhere here the yeah, so they included moles as well. So microbiology is actually defined in the terms of its culturing techniques and its classification. Wait, there was something else that was written here. Microbial classification. Anyway, so in techniques, here it is. In technologies such as sterilization and the use of culture media. All right. Sterilization is the completely, killing of the microorganisms, and culturing is the growth of microorganisms in a required media. Like these two are the ways, like how do we grow the microorganisms and how do we kill them. Like this completely embodies the techniques, like every other technique is in between these two. So describe and contrast prokaryote and eukaryotic cells. So first of all, we know prokaryotic cells don't have true nucleus or double bonded, organelles, while eukaryotic cells have the true nucleus, which is surrounded by the nuclear membrane, and they have more complex organelles than this one. So in prokaryote, in microbiology, we have bacteria and archaea, while in eukaryote we have all the remaining like algae, fungi, protists, like every other that's remain is under the eukaryotic cells. So the contrast is just that simple, that it doesn't have true nucleus while this one has true nucleus. Describe and contrast the five kingdom classification with the three domain system. Why do you think viruses are not included in either system? First of all, this five kingdom classification system is done for the living things, right? And this three domain system is also done for the living things. Viruses are not considered living. They are at the borderline of the living and non-living. So if they are not even considered living things, so we don't classify them in these five kingdom classifications, right? Like we can say if this is a prokaryotic cell or if this is a eukaryotic. This isn't even cell. This is acellular organism, right? I don't know if we can call it an organism as well. This is a molecule, just like a genetic material surrounded by the protein capsid, or few tail fibers or something, and these few enzymes in them. So my reason for them not included in the five kingdom or three kingdom classifications is that they are acellular and that they are not considered living things, right? Now comes the five kingdom classification and three domain system. So first, they generally classified the organisms into three domains. Here they are in the diagram: eukarya and the prokarya. You can say the prokarya and the eukarya. Prokaryotes means not true nucleus, and in those we have bacteria and archaea. So they classified them into three different, different. In eukarya we have animals, plants, I think they are, yeah, homo, yeah, here you can see homo sapien, that means it's animal. So in eukarya we have animals, plants, and algae, protists, fungi, all of these kind of things. So but in prokaryotes they are classified further, and they are bacteria and the archaea. Bacteria, these two are different from one another, bacteria and archaea, by their ribosomal RNA. Their ribosomal RNA is different, and so that's why they exhibit different characteristics. First, in the ancient times, they weren't considered different. They were just considered as single bacteria, right? But after studying their ribosomal RNA, they made sure that they are completely different organisms, even though they look the same. And both are, classified into further classes, and they both have a lot of other classificated organisms from them. So this was the three domain system, right? But then later they found out that this eukarya that we made into one kingdom, they thought that this was way too general for them. Like this, there was less specification in this, so they decided to separate it, and I think they combined the prokaryote, the bacteria and archaea. Like they thought, why not put them together? They are more diverse than these two, so they put them into one. And wait, there it is. Here it is written somewhere here. So the 5 kingdom classification that we are very familiar with are these: Monera, Protista, Fungi, Animalia, Plantae. So Monera had the bacteria and the archaea, the prokaryotic cells, right? Protista had the protozoa, protist, like these kind of things. Fungi, we know fungi, the multicellular or unicellular eukaryotic cells. And the Animalia, we know animals, and the Plantae, we also know plants. Sorry, not animals. So these were the 3 classification system, right? Except for viruses, which are acellular and have their own classification system. Yeah. Microorganisms were placed in the first 3 kingdoms. So they are saying microorganisms are included in these. Like I said, the Monera has the bacteria and the archaea. Protist has the algae and protozoa, while this one's fungi. So these are all the 5, excluding the viruses, which we study in detail in microbiology as well. So this is our answer to that. So next one are these 2. I don't know what these are. I hope I get the answer for that. So give some example of the kind of information you think can be provided by the microscopic observations of the microorganisms. Alright. So they are saying if you study the microorganisms under the microscope, what kind of observation, like what kind of information can you get from just looking at them morphologically, outer appearance? So I've studied that in detail. I don't know if we studied this in chapter. There is the color, like first of all, microorganisms, if you talk about the bacteria, right? So they grow in colonies. So colonies have their own characteristics, like first of all, their color. They are whitish, creamy, black, gray, brown, yellow, orange, and everything in between these colors. They can be that color, right? The bacteria. In fungi, we usually study the hyphae, the segmented hyphae or unsegmented hyphae. We study the spore, like their shape, their size. Oh yeah, in colony we don't just study the color of that, we study their size, their thickness, their leanliness, their concaveness, their convexness, like these kind of characteristics as well. Their smell, all of these. And fungi, yeah, we study these hyphae and spores and segmentation and non-segmentation, and yeah, color as well. While viruses, we usually see the what do you call it, the bodies or the morphological effects that it causes in the cell. Usually the microscopic observation cannot be done to the viruses, right? Because viruses grow in the cell. Well, we can study that cell under the microscope, but we cannot study the virus directly under the microscope. And like we can study that by through the scanning electron microscope or the what do you call it, one is scanning and what is the transmission electron microscope. So we can do that with that. Again, we study the morphological, like the spikes and the color, but like I said, viruses are usually characterized by their antigen antibody kind of things, not we don't usually diagnose the viruses under the microscope. Again, now we comes to towards the algae and these kind of things. I don't think they will need the microscope, right? Like we can study them unless they are unicellular or they are microscopic. Algae are usually grown, in a colony, like we can see it with the naked eyes. And same case, with protozoa as well. Like, yeah, these kind of things. Usually what we study under the microscope are bacteria and fungi mostly, right? Like we can study the protozoa and algae, but I am not that much, familiar with that. Give some and neither have they explained this in that chapter. So give some examples of the kind of information you think can be provided by isolating microorganisms from their natural environment and culturing them in the laboratory. Laboratory or laboratory. These are 2 different pronunciations. So they are telling me to give examples. What kind of information I think I can get by isolating the microorganisms? What happened? From their natural environment, culturing them. What kind of information? What do they mean with kind of information? Like I said, I will know their colony shape, their colony size, and what class they do belong to, if they are familiar with the some kind of other microorganisms. And they are not talking about just the what do you call this, not talking about just the microscope, right? We can do the biochemical tests like catalase tests, like if they produce oxygen or not, like if they produce, yeah, H2O, O2, H2O2, I think that's what the gas that is they produce. So or if they live in aerobic conditions or anaerobic conditions, like we can separate them. And most important thing which I forgot to mention in the last question is the gram staining. Gram staining is, like, we usually divide them into the gram positive bacteria and gram negative bacteria because of the staining. Like if they retain the stain, that is gram positive. If they don't, they are gram negative, and we further stain it in a different color. That's a different explanation. I think I explained it in the previous lives, so we don't have to go. So yeah, by gram staining we can isolate, we can differentiate them if they are gram positive and gram negative. And believe me, gram positive and gram negative bacteria are so different from one another. Their characteristics just different. You can say they are the main two classifications under the bacteria, right? So we can do that as well. And examples, I just, what do I do? I should I type this in a Google and ask the Google what kind of examples do they give? Because I don't have specifically any example in mind, neither have they explained it in this chapter. So let's copy it. um, what the heck is this? Wait, did I read the question wrong? Give some examples of the kind of information you think can be provided by from. Okay, yeah, they are talking about the information. Yeah, the kind of information. Yeah, 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 yeah. I already explained. I don't think we need to go dive deeper into that. Like we are not some kind of experts and we can't provide answers from our own which we haven't even studied. This is just the first chapter, right? We are on our way to study more. So here there are no more questions for now. We are going down. Here. Where am I going? Here. How did Pasteur and Tyndall finally settle the spontaneous generation controversy? So, anyone watching this for the first time, is spontaneous generation. We studied, I think, whole 40 minutes the spontaneous generation, and that was that life. It was the controversy that life comes out of nothing, and then when it was solved for the macroorganisms, they were still stuck at the microorganisms level, that they still thought that the microorganisms could come out of nothing, right? And then there were scientists, like they were experimenting one by one, and we have already studied all the scientists to to kill the spontaneous generation controversy. Yeah, so the last two scientists which were included in the finishing of this spontaneous generation controversy were the Pasteur and Tyndall. Tyndall first and then Pasteur. And Pasteur is a big scientist in the microbiology. Like he did a lot of things. I think you studying this chapter with me in the lives, you will know how with how much did he did. So here it is, Tyndall. So he demonstrating that dust did indeed carry organism. Yeah, so he finished the spontaneous generation by experimenting and explaining that dust had germs, like germs were the cause of the life that came out of the meat, or you can say the organic matter that were left. It wasn't air or neither the organic matter, both were heat killed, but there was something in the air or something in the medium that would give rise to the germs. So he provided that existence of, yeah. Another thing he did was the existence of exceptionally heat resistant forms of bacteria, but this isn't related to the question. Like they just said how did they settle the spontaneous generation controversy, and he settled, Tyndall settled it by telling, give me a second, So Tyndall settled the spontaneous generation by telling that the germs were the cause of the generation of the bacteria or the sprouting of the bacteria. It wasn't out of nothing, but Pasteur did multiple experiments, series of experiments, and he proved that yeah, these were the microorganisms. And yeah, again, wait, so Pasteur was the first one. I am sorry for the mistake. Pasteur clarified it first and then Tyndall, as said in the question. So, okay. So Pasteur did multiple series of experiment as well. John Tyndall just further proved that yeah, Pasteur was right. This is Pasteur. Now this is Leeuwenhoek, right? So the Pasteur boiled the solution. So he sterilized the solution as well, and he sterilized the air as well. And how did he sterilize air? He made these, looking like these kind of flasks that had curved necks, and the air wouldn't go to the boiled sterilized medium, and in this way no growth of microorganisms will occur. But then he cut these curved flasks and the microorganisms grew in the media as well. So that he said that it wasn't the air, it was the particles or the germs that were left out, settled down at the necks of these flasks, that were the cause of the growth of the microbes. So this was that, and the Tyndall was, yeah, just as I said, that there was germs in the air. He named them germs. So why was the belief in the spontaneous generation an obstacle to the development of microbiology as a scientific discipline? Like, belief in the spontaneous generation was an obstacle? To me, it wasn't an obstacle. I think it was a, it was a, it was actually a stepping stone for the microbiology to go further, because people were actually started experimenting on the microorganisms, the same microorganisms which they couldn't believe till now like they existed or something. So the spontaneous generation, when said that the microorganisms come out of nothing, so they had to experiment on them, and they have to know what actually is the thing that is leading to the growth of microorganisms. Even though they didn't know how to culture these microorganisms, they discovered the technique that was sterilization, like through boiling, that they could kill the microorganism. So they developed the sterilization technique first, like the Pasteur proved that yeah, boiling can kill the microorganisms if provided the specific temperature, right? But after that, they did experimented on them so many times that this is all the reason for the spontaneous generation, that the microbiology has advanced to such degrees. It was the start. Like microorganisms, no, sorry, scientists were obsessing with, like, there is no way life could come out of nothing. There must be some reason for that, right? Scientists usually, they say they don't have religion. Science don't have religion. They try to seek answer in everything. So the idea of microorganisms coming out of nothing or organisms coming out of nothing is absurd. So they experimented on it. So I don't consider it an obstacle. I think it was a stepping stone for the microbiology to start, because it was by this time, like starting with the Tyndall, right? Tyndall, like he found out the heat resistant bacteria, and then the Ferdinand Cohen, he found out the bacterial endospores. So this was the start, like, yeah. And then people started believing as well that yeah, the microorganisms were something that continue to reproduce, and they exist, like they aren't something that come out of nowhere whenever they want, right? So when they found out that yeah, there is a reason for their growth, so after that they started to develop the culturing techniques and things like that to actually reproduce the microorganisms over and over again. So let's go to the next questions. Yeah, so here are the lots of questions. So Lister, Pasteur, and Koch to the germ theory of the disease. First, you have to know what is the germ theory. Germ theory of the disease is that the microorganisms cause the disease. In here, the germs means microorganisms cause the disease, and that disease can be transferred to another person if we put the same microorganisms into another person. So not person, it can be any living creature, right? So they are saying what were their contribution. So Koch obviously had the most contribution to the germ theory. Why? Because the Koch postulate, he actually proved it step by step, and we already studied in a previous live. Here you can see, I'm not talking about the molecular postulates because it's something else. And yeah, it is related to the germ theory, but in here they are talking about this one, like the four postulates, right? You can culture the microorganisms from unhealthy organism, and then they grow, and yeah, they can grow in a pure culture, and then you want to inject into a healthy animal, they can, organism, you can they develop the same kind of symptoms and diseases as the previous one. And when you take out the sample from the unhealthy organisms and you grow those microorganisms in a culture, they are exactly same, identical to the microorganisms that you first took from the first unhealthy microorganism. So that was kind of Koch's, demonstration of the germ theory by making the Koch postulate. So that was that. And like, he found out that anthrax bacillus was the cause of, anthrax disease. So you can say, in kind of sense, he also proved the germ theory. About Pasteur, as you can see, wait, did Pasteur did something in a disease area as well? Wait, yeah. Oh yeah, Pasteur, by growing it an abnormal host, and then he died. Oh yeah, this story I completely forgot. Yeah, so this was the Pasteur did develop the rabies vaccine, right? We know that you can, take the microorganisms, like microorganisms, or you can say in this case, yeah, in this case, wait, it's a virus, right? It's a virus. So in this case, he developed a vaccine, and the vaccine was developed, like, killed, like vaccine, he killed them, right? This incubating the bacteria. Yeah, that's an anthrax vaccine. Yeah. Yeah, sorry for my network, it's stuck. Let me check if it is working fine. Wait, let me, no, like, I don't want things to happen. I need to know if this is working fine, because my OBS suddenly disconnected and I don't want it to go wrong in any way. Let me see. Wait. This is working fine. A 9-year-old boy was bitten by a rabies, virus. A dog that was, the reservoir for rabies virus, and then when he injected the vaccine, so he showed that it was, killed. So it was the development of vaccine in which he contributed to the germ theory. And this lecture, he developed the antiseptics, you can say, for the surgeries which the doctors do. And he said that it could prevent the infections by the microorganisms. Like, it is also a proof to the germ theory of disease that, yeah, microorganisms cause disease. So, and how did he prove that? When he provided these antiseptics on the injuries that he was, operating or things, the operation did succeed, like, and in this case it was the phenol antiseptic he used. So the surgeries were a success after his, antiseptics, and they call it antiseptic surgery. So this was Lister's doing. Then what contributions did Koch made to microbiology? So Koch did find out that the anthrax bacillus was the cause of the disease or infection, the anthrax, right? And he did by culturing them and this and that. The whole story is in this, here. Yeah, in the beef serum, and the bacilli grew, and then he injected into mice, and the mice developed anthrax as well. And after that he did the same case with the mycobacterium tuberculosis, right? The disease, the infection that was happening because of the mycobacterium tuberculosis was the tuberculosis, and he did step by step the same steps he did with the anthrax as well, and from that he formed the Koch postulate as well. So first this was his contribution, but when you advance into that, so there is a Koch molecular postulates in which he said that the microorganisms have some virulence factor, pathogenic factor that is associated with their pathogenicity, right? And if you inactivate, and there is a gene responsible for that pathogenic factor, if you inactivate that gene, it won't be that much pathogenic as before. And if they somehow gain the same gene, a wild type gene, their original gene, from environment or from another bacteria or from any other source, they will develop the pathogenicity again, and antibodies or immune system direct against the gene. So the gene that is responsible for producing pathogenic traits, our immune system actually targets those pathogenic factors to protect hosts. So these were general contribution. And after that, when he was culturing the mycobacterium tuberculosis, he had struggling with the making of the medium. For the medium is a material which is a complete nutrient to grow the bacteria. So he was having trouble with developing that, and in this case his assistant's wife or something, Fanny Alishmas and the Walter Hesse. So she said that in her work with doing research, she used the jelly, so agar, you can say agar was that jelly which you usually use these days for the solid media. And when he used that, the bacteria grew quite nicely. So Koch also, you can say, kind of normalized the use of the jelly, or you can say solid media or the agar media. So describe the Koch postulate. What is pure culture? What are pure cultures? Why are pure cultures important to Koch postulates? I already said about the Koch postulate. I don't want to repeat it. Pure culture, is a growth, is a colony which has only, can be a pure culture of anything, can be a pure culture of viruses, can be a pure culture of bacteria, can be a pure culture of fungi, anything. Like you have nothing other than what you want on the media you are growing them on. So this is pure culture. And why are pure culture important? Of course it is important because we want to have a pure culture of the organisms that we want. What are we going to do with a mixed culture which have different microorganisms that we don't even need? And you guys can know that different microorganisms affect wanting the right microorganisms. So pure culture is important. Would microbiology have developed more slowly if Hesse had not suggested the use of agar? Give your reasoning. Would microbiology have developed more slowly? I think yes. They are asking for my opinion. Fanny is the wife of that assistant, right? The use of agar, because I think if you look into our laboratories these days, the agar is the common type of media that we use for the growth of. If we don't have the agar, we usually go towards the other mediums and things like that. But people that are not expert, and agar is just such a basic nutrient source for the bacteria. It is solid, it has temperature, it doesn't have temperature problems. So sooner or later the scientists were going to develop the agar in one way or another way, but that is left to the fate. If the scientists would have found this agar sooner or later, I can say if they have developed more slowly. So it all depends on if they found out the use of agar, because we don't have that much alternatives for the agars. You can say like that works the same as agar, right? Give your reasoning. Yeah, I told you my reasoning. Like, because it doesn't have problems and such a quick source of nutrients instead of serums, right? Like, there are serums that can work the same way, but serums have so much condensed nutrients that bacteria, I don't think every bacteria can survive in the serum nutrient, and it is difficult to handle the serum media. So yeah, I think it would have developed slowly if they didn't found the agar even after the Fanny Hayes. The scientists would have come to this conclusion sooner or later. What are the Koch molecular postulates? Why are they important? They are important because this led us to the finding of, the pathogenic factoring of the microorganisms, and what are the factors of them causing diseases. The microorganisms could have one pathogenic factor or multiple. So Koch molecular postulate, I'm already done with this, I'm not revising it. Some individuals can be infected by a pathogen yet not develop disease. Okay, so they are saying that some individuals can be infected by a pathogen, but they do not develop disease. In fact, some become chronic carriers of the pathogens. How does this observation impact Koch postulate? How might the postulates be modified to account for the existence of chronic carriers? So, in my case, the individuals that are infected with pathogens, right? Chronic carriers means if the same pathogens go into another human body, they could cause a severe disease, but in the first individual it isn't causing disease. They are saying how is it related to the Koch postulate? For you can say if the microorganisms is for attacking the dogs, and it is found in human, it is a reservoir of them, and it infects the dog, it will cause disease in dogs but not in humans. First is this. The second is it may be because the individuals that have pathogens in it, their immune system is so boosted that the pathogens doesn't, cannot cause diseases. And usually when the individuals are immune to some pathogens, they kill the pathogen. The pathogen don't live in their body, right? Not for long, because our immune system are quick, they can left foreign particles in our body. So in that case, if as soon as the individual was infected and in their body they transfer it to another person that's immune system is weak, they are going to get disease or infection from those microorganisms, unlike the first one. But I'm not sure how Koch postulates are related to this. Koch postulate didn't tell us anything about this. Yeah, like this is not in the Koch postulate. This is the limitation of Koch postulate, right? Koch postulates told us that if we take the microorganisms from unhealthy organisms, right, they are going to cause disease and other organism. Like, but in this case there is some super individual that came here, that immune system is either so strong or species is different, then the Koch postulate doesn't apply because it isn't going to cause a disease in that organism, and maybe the microorganism doesn't even survive in that organism because of their immune system. So the postulates can be modified, like you can say like the the organism should be the same species, the organism should have the same immune level as the one that were infected first or that's going to be infected after. So I don't think this much modification will work. Yeah. So describe the scientific methods in your words. How does a theory differ? If I'm wrong in the previous question 6, correct me, because I think I can go to that level for now. A scientific method in your own words. How does a theory differ from hypothesis? Why is it important to have a control group? So scientific method in my words. Scientific method is you take observation, you make your hypothesis, like you make a statement of what you see, a problem. You have a problem and you make a statement of what you see, then it becomes the hypothesis. Then you take the observation of from your surroundings, and then you see and check if your hypothesis is correct or not. If it isn't correct, you are going to make another hypothesis. If it is correct, you are going to make experiments on it more and more. First you took observation, now you are going to experiment to check out if it is right and wrong. If the experiments are okay, then your hypothesis is okay. If it goes wrong, then you are going to make another hypothesis, and this steps follows and follows until there comes a point where there is no other way, like from no other angle you can prove that your hypothesis is wrong, and that becomes the theory. And if you make the theory experimented on from every angle, from math, science, physics, every kind of, I mean, like whatever you can do from that, and it still is stuck in the same place and it doesn't budge, your hypothesis is just that strong, it becomes a law. Why is it important to have a control group? Control group is the one that we know what the answer is, right? Like you can say the teacher have a paper and the answers are all written there, and we know what the correct answers are and we know what the wrong answers are. Like this is the control group. And then there is a class like which have the papers, and then some people get the right answer, some get the wrong answer, and then we compare it to our control group, like we check it. So the same case is with the microorganisms. We check for the presence and absence of microorganisms. Like I'm giving example. People say this guy is HIV positive and that one is HIV negative. How do we know which one is HIV negative and which one is HIV positive? Because we have a control group. We already did experiment on one thing and we know what we do. What will we do to make it come either positive and negative? Like we know if we put this, it is supposed to come positive. If it doesn't come positive, that means it's negative. Like the control group is necessary for our experiment to be set in place. Like we can compare it with the right thing. Like if we don't have something to compare it with, we might come across some mistakes. Not some mistakes, humongous mistakes. How did von Behring and Mechnikov contributed to the development of immunology? Mechnikov, shown here at his laboratory. I think Mechnikov, well, Behring did discover the viruses like tobacco mosaic viruses, and he did all the procedures which we already studied in the lab. While Mechnikov, I forgot what he did. Huh? What did Mechnikov do? Discovered some blood leukocytes could engulf. Oh yeah. Phagocytosis. He discovered the phagocytosis. Like we knew that there was something in our immune system that could assist our immune system, something in our immunity that could assist the immune system in working strongly. And that were the humoral immunity antibodies, that was that. But after that they found out that blood leukocytes, you can say white blood cells, are also responsible for immunity, and it is done by the process known as phagocytosis, right? The phagocytosis occurs in the macrophages. I don't know. So Behring is Behring, here it is. Oh, it was Behring. Oh, sorry, the tobacco mosaic virus was discovered by the Behring. I mixed their names. Behring and this scientist Kita Soto was actually responsible for producing antitoxins against toxins of rabies, not rabbits, against the toxins of what and what. There were two diseases that they produced antitoxins for. Yeah, here, a tetanus and where is the other one? Diphtheria. Yeah, for the diphtheria and for the tetanus causing microorganism. Briefly discover the Pasteur's work on the microbial fermentation. So here is the story of Mr. Bigot. He was doing fermentation with beet sugars, and for some time his beer wasn't producing, in a quality which he usually had, and he came to the pastor and asked him what the fermentation, what's wrong with his fermentation. And so the pastor told him that there was some kind of contamination in his fermentation process, like there were some kind of other microorganisms involved with the microorganisms that are needed for the fermentation of beet sugar. And so he changed his temperature, pH, and this kind of things for the process, and then his beet sugar fermentation produced the liquor, or you can say beer, which he usually would produce. And of course the pastor was the first one that found out that the microorganisms, or you can say he told the people that microorganisms were the ones responsible for doing fermentation. Winograyski and Beijering contributed to the microbial ecology. Like I told you, Beijering was the one that discovered the tobacco mosaic virus. And this guy, Winograyski, to soil microbiology by discovering a lot of soil microbiology, which iron, sulfur, ammonia, oxidizing bacteria, and he also discovered the photosynthetic organisms, photosynthetic bacteria like cyanobacteria. He also found out the anaerobic nitrogen fixing bacteria as well. While I think the Beijering also discovered the aerobic nitrogen fixing bacteria. No. Yeah. Beijering discovered the aerobic nitrogen fixing bacteria, and he also found out the azobacter, bacteria that lives in the roots of the plant's root nodule bacterium. And later it was named as rhizobium, which is also responsible for fixing nitrogen. And he also discovered the sulfate reducing bacteria. And the biggest thing which these two did was the development of enrichment culture. Like this is a specific type of media in which we enhance some nutrients and decrease some to make it selective for some organisms and not selective for other organisms. Like for halophiles, the salt loving organisms, we make the salt quantity in the nutrient media more, and so other than halophiles don't grow in there. Leeuwenhoek is often referred to as the father of microbiology. How many historians feel that Louis Pasteur, Robert Koch, or both deserves that honor? Who do you think is the father of microbiology? Like, father of microbiology is a title that was given to Leeuwenhoek. Either it is like, be it by chance or be it by anything, Leeuwenhoek was the first one to discover the microorganisms, and he reported that. So father of microbiology goes to him, even though he did nothing else than that in this regard. Even though Louis Pasteur, Robert Koch did a lot more than Leeuwenhoek, not by few 1-2 scales, by hundreds and thousands scales. Louis Pasteur and Robert Koch did a lot of them, but it's that he's the founder of bacteria. If he didn't found the microorganisms, maybe someone else might have found the microorganism, and then that would be the father of microbiology. So the father of microbiology, or father of anything, is the one that will produce the result of something. So it's Leeuwenhoek, be it by chance or by anything. And Louis Pasteur and Robert Koch deserve that honor? They don't. Why will they deserve the honor? Like, they are not the first one to, or you can say they will become stepfathers. But that would just mean that all the all the microbiologists till today are the stepfathers of the Leeuwenhoek. Not the microorganisms, right? Microbiology. So I don't think they deserve honor. They don't deserve honor to be the father of microbiology, but they do deserve honor for the work they have done in the field of microbiology. So father is a different thing. Father doesn't mean, like, in my mind, Robert Koch did more than Leeuwenhoek, but Leeuwenhoek was the first one to discover the microorganisms. So, well, there goes my reasoning. Consider the discoveries described in section 1.2 to 1.5. What do you think were the most important to the development of microbiology? What? 1.2 to 1.5. Hey, they are telling me to revise the whole chapter. I can't do that. The important discoveries. Everything was important. 1.2. So it started with the micrographia. It started with the living hook, so he discovered the microorganisms. That is an important discovery. And then the Pasteur debunked the spontaneous generation theory. That was important. And along the way, the agar was no, sorry, that was just debunction. So yeah, so all of these scientists are important, but I think the most important thing is that the Pasteur debunked the spontaneous generation as a set stone, and then the Tyndall and the Cohn discovered the heat resistant forms of bacteria, or you can say the endospores. Then they made the vaccines as well. They made the they made the Koch postulate as well. They found out the fungal infections like the rust and smuts. They found out the silkworm infection as well. They found out the surgical antiseptic surgery, Koch postulate. All of this is important. I don't think anything is less important than the other. Especially it is because it's the early age of microbiology, so every single thing matters because it all comes in the basis. Richard Petri discovered the Petri dish. Well, to me, it's the least important one, but I think it's the most important one. All the microbiologists or the scientists studying or growing the microorganisms use the Petri dish. The development of industrial, yeah, everything is important till what we studied till now. So, is there any other question remaining? No, nothing. Alexander Fleming, we completely forgot about the Alexander Fleming, which he discovered the first antibiotic that could kill bacteria. Like, there was no specific phenols. There were chemicals that could kill bacteria. There were methods, techniques like boiling, pasteurization to kill microorganisms, right? But antibiotic, that was the first one to be produced by fungi. Number 1, briefly describe the major disciplines in the microbiology. Guys, everything related to microorganisms is the subdiscipline in the microbiology. Bacteriology, virology, algaeology, fungiology, mycology, microbial ecology, food microbiology, industrial microbiology, microbial ecology, everything are the major. Although major are the just 5, I think. Bacteriology, virology, algaeology, mycology, and the protozoology. These are the basics. And after that are the basic disciplines are the immunology, the genetics, the microbial ecology, industrial microbiology. Well, there are a lot of them. Why do you think microorganisms are so useful to the biologists as experimental models? Well, they are small. They are easy to grow. They are easy to manipulate. They are easy to give nutrients to. They are easy to handle. They are easy to produce the desired products which we need. We can insert whatever gene we want, well, at simple ones, and the things they produce will be in large quantities. So yeah, that's why they are useful. And we can study them under the microscope. We can change their, we can make their nutrients and the environment as we desire in the lab as well. List all the activities or businesses you can think of in your community that are directly dependent on microbiology. Activities and businesses. I don't know about the practical side of microbiology in my community. Directly dependent. First of all, it's food. Yeah. The canned food or the food that comes in packaging. That's directly related to microbiology because they are being pasteurized either by spices or by techniques. The milk is also pasteurized by high temperature and pressure to kill microorganisms. The fridge works the same, the freezer works the same, either by, yeah, it's by biostatic. The freezer and the fridge is biostatic. They don't kill bacteria or viruses or microorganisms, but they do stop their growth for some time. The spoilage is also because of the microorganisms. The water contamination, the diseases, the infection, the diarrhea also are related to microorganisms. Is there any other question that remains? What do you think are 5 most important research areas to pursue in microbiology? Give reasons for your choices. Huh? If I knew that, would I have had this much problem reading the book? They're really asking me this question. 5 most important research areas. So the first is medical microbiology. Like it started with this, the infections. The microbiology advanced in the medical field the first most because it is related to human health and human survival. So that's the first. And by the way, if you let these medically important microorganisms free, it will cause disaster. And simple as that. There's a coronavirus and there was the black plague in the history that wiped quite the population of the earth. The second is industrial. Yeah, industrial microbiology has dominated like that kind of area, like the food and the manufacturing of insulin, medically important antibiotics, and all of these things are produced in industrial microbiology. What is the other one? Ecology. Ecology, really? Ecology. I think immunology or biotechnology is more important. Yeah, for the advancement in microbiology. Like if the first industrial microbiology and the medical microbiology are important to us, the genetics and these are, you can say, help in the advancement of microbiology or the worlds that we live in. If we know the genetic manipulation and the genetic studying of the microorganisms, we can then go towards the higher organisms. And we already use the bioengineering in the microorganisms to produce our desired product or to change the characteristics of the microorganisms. Here is a gene therapy. I think it's also done by bacteria or something. They not bacteria, by the viruses. Yeah, and the other one is cancer. What do you call it? Cancer microbiology? No, I forgot about it. So 5 most important. How many did I say? Medical, industry, genetics. Immunology is important because infection is the usual thing that activates our immune system, other than the allergens and the pollutants or the hypersensitivity or the autoimmune disease. Other than that, all of the reason for the immune system activation is the microorganisms, and the whole system is made just to fight off against these microorganisms which we can't even see. So the more we study our immune system, the more we study the action of microorganisms, the more we will be capable of developing antibiotics or vaccines or boosting our immunity or studying the susceptibility of microorganisms. So that is the immunology. Ecology, should we study about the ecology? Yeah, so microbial ecology is also important because the food that we take comes from the soil, and soil has how much microorganisms? 99%? No, sorry. I think most of the population of the microorganisms are either in the living being's body, and the first one is the soil. It's the most abundant microorganism reservoir in this world. So they have bacteria from starting layers to the under layers to the deep layers in the soil, and they all have different functions. Yeah, why is not ecology important? What am I saying? If there wasn't microorganisms, who would change the nitrogen gas in the environment to the ammonia and the nitrates and nitrites that plants use? If they couldn't convert, there would be no plants on earth. We know that the sunlight and the oxygen, carbon dioxide is necessary for the plant's photosynthesis, but where do they take the nutrients for them to grow? It's from the soil, and they have the most important is the nitrogen. If microorganisms weren't there, they wouldn't be able to survive without that nitrogen. Even the fertilizers have the nitrogen and the sulfur capacity the most. So yeah, microbial ecology is also important. So to me, these were the 5 most important research. For now, that comes to my mind. Other than that, I can't think of anything. So the remaining chapter I'll just deal with future of microbiology. For now, we are done with the question answers. So that was it, and I'm ending the stream.